draw a video, I can only draw one thing at a time. So what we're looking at, the lumen, I actually draw this the reverse, but the lumen is down here. This would be the surface, the simple columnar epithelia of the surface of the uterine cavity. And these are uterine glands, lined with a simple columnar epithelia as well. And these are branches of the spiral arteries, because the source of nutrient for the developing embryo and fetus, of course, is maternal blood. So here we have the blastula or blastocyst with the hollow ball. This is already implanted in, but we'll see it processed. And we'll talk about the cells that are involved once this video is over. That's not what I wanted to do. And this does not have sound either. You can see it's come through the oviduct, the isthmus of the oviduct. It's now moving down the uterine surface. It doesn't eat its way into that stratum functionalis. It's actually going to kind of release chemicals that cause the cells to separate rather than break down. And the single layers of cells are known as a trophoblast. They start to multiply and fuse, forming a syncytial trophoblast. So the individual cells are now called cytotrophoblasts, and the fused tissue a syncytium. Eventually, the maternal surface of the uterus will come back over and cover the blastula. As it sends out finger-like structures of syncytium, it actually invades the maternal blood vessels and sends fetal tissue up to line the maternal blood vessels so that they're in direct access with maternal blood. So we'll look at the beginning of this process today. We'll review and finish it on Tuesday, and then look at what happens. This is our developing embryo right here. All right, so let's look at those steps, and you have them in your lecture notes. I think there's discussion of it on page 134, and the pictures are that I will show you on the board are start on page 138. I printed mine off without lines, so when I first gave pages, I was off by about 70 pages.
What are you looking for?
So I'm going to draw mine with the lumen up here, all right, to get you oriented. What happens, and let's just do a preview here. So we have the uterus, and I'm not going to draw it in its anatomical position. Here would be the cervix. The blastula comes in, settles down, and implants underneath, through, and within the stratum functionalis. So this is not a cell. This is the blastula with its inner cell mass. Okay, it's going to develop its placenta from that structure around here. But it's covered, the surface epithelium of the uterine cavity grows over it and covers it. As this develops then, it bulges that surface epithelium into the uterine cavity, and so we start to see this. So this is the epithelium, and down here would be the stratum functionalis. And our placenta, the embryo will be in here. And then as it gets larger and larger, it continues to obliterate the uterine cavity. So it's always within the stratum functionalis. So this space here was that original uterine cavity. Right? So technically, the developing embryo and then fetus is never actually in the uterine cavity itself. It's in the stratum functionalis, covered over by the surface. And then eventually, they never actually grow together, but this becomes a tight opposing layer. And we have our cervical canal and structures like that. So on Tuesday, when we come back over and finish this implantation, we'll talk about what tissues then have to be, if a doctor decides that a woman's amniotic sac is not going to tear on its own, the rupture of the water, if you will, what tissue, when he takes that, or she takes that crochet hook-like type of instrument and does it themselves, what layers is it going to go through? Okay, we're not going to get this far today. We'll get the blastula into the stratum functionalis and covered over and see the formation of the early fetal and maternal tissues, and then we'll come back, review that on Tuesday, take it further, and then also talk about the first few weeks of fetal development. That's the placenta. That's the fingers of fetal tissue that are growing into the maternal tissue. I'm going to diagram it. I was more interested in this part here, so I wasn't drawing that quite as clearly. All right, so on the first image out here, you can see that the blastula or blastocyst, either term is correct. So the morula is the solid ball. By the time it enters the uterine cavity, it's a hollow ball of which the outer layers of cells are in a single layer. And then we have a cluster of cells on one end. These cells are identified at first as just trophoblast. And these cells form the inner cell mass. The inner cell mass is going to become our embryo and our amnion, or the amniotic sac. The trophoblast will become the placenta along with maternal tissue. What would be the chromosomal pattern of our trophoblast cells? 2N. Good. All right, so I'm going to erase this diagram and redraw it.
Hold on, tropo. Okay.
Okay?
But then the ovum is in the inner cell mass somewhere.
The ovum is no longer an ovum. The ovum was fertilized to become the zygote, and now its chromosomal material has its further divided. It's gone through hundreds of cell divisions and is forming each of these. And it's a round cell, so it tends to look like it's a cellular tissue. Pardon?
At what stage does it become embryo? From the...
Well, once we form, I'll show you. Well, we won't get to it till Tuesday, but from the inner cell mass, that will separate to form our embryonic disc.
Oh.
And then we'll talk about that and the folding of the disc on Tuesday. We're not going to go through all 9 months of development. So here's our lumen with the uterine tube, the uterine glands. And blastula, what colors I want to use here. Has already, these are simple columnar cells. That's our lining epithelia of the uterus. And our blastula has already separated some of those. And is settling into the connective tissue of the stratum functionalis, the most superficial part of our endometrium. So as such, this is still trophoblast. We haven't yet formed our syncytial trophoblast. Okay. So it settles down. We have our inner cell mass here. So partly on the stratum functionalis, partly still in the lumen. the trophoblast cells start to multiply. They undergo mitosis. And they form multiple layers of cells. And some of those multiple layers of cells fuse together. They lose their plasma membrane, dividing, indicating, separating each as a separate cell. So I'm going to draw that fused tissue. It's shown here in the lavender. I'm going to draw that fused tissue as red. So these were the trophoblast cells that lost their plasma membrane specificity and now share the cytoplasm and nuclei as one large, kind of like a fungus, one large living organism.
Do we have a name on ourselves?
So the cells, the individual cells, we now call cyto, which means cell, cytotrophoblast. And the fused plasmodium component, syncytial trophoblast. Okay, before we could tell the difference, but it was just the blastula, we just call them trophoblast cells. All right, once they fuse and form a syncytium, then we can divide it as cytotrophoblast and syncytial trophoblast.
What's the definition of syncytium?
So syn means with, so a syncytium is the fusion. It's just the large cytoplasm of multiple cells within one structure. This continues to move deeper into the stratum functionalis, and as it does so, more cytotrophoblasts are formed and fused and become part of the syncytium. All right, so the cytotrophoblasts continue to divide, and as they do so, our syncytium is growing and sending out those ruffly things we were asking about, and penetrating farther and deeper into the stratum functionalis. As such, it causes a chemical change and structural change in the fibroblasts of the mother that form the stratum functionalis. That change is known as a decidual reaction. And so we now call the stratum functionalis of the entire uterus the decidua.
Can you spell that?
Okay? I will. So the decidua is the connective tissue component, or just the tissue here of the stratum functionalis, all the fibroblasts and all that other. It's an inflammatory reaction, a swelling of the cells that occurs. There's a dearth, a disappearance of our immune cells, because half the proteins on these fetal cells, or the trophoblast cells, came from the father, so the mother doesn't recognize them. And in some cases, mounts an antibody response and doesn't allow implantation to occur, rejects it. So I'm going to show the next stage here. So now this is completely submerged beneath the uterine epithelium. So I'm going to erase this and redraw it all the way underneath our uterine epithelium. So as it's moved deeper and deeper and deeper, the uterine epithelium has closed over the surface. Here's the lumen of the uterine cavity. Here is our blastula still. Inner cell mass. We're going to come back and do the inner cell mass changes on Tuesday, so I'm not going to, even though they're separated and form several layers here, I'm not going to worry about that. Our cytotrophoblasts. although our syncytium is getting larger and larger with every day, the cytotrophoblast still remain. All right, so when I was doing my doctoral research, I worked on a cancer of placental cells called choriocarcinoma, but I looked at non-cancerous cytotrophoblast by staking myself outside the delivery room at Sutter Memorial, and if they had a C-section, they would get the parents' permission to take the placenta, put it in an ice cream container. I take it back to the lab, put on safety glasses, go snip, snip, snip with scissors, and use digestive enzymes and centrifuge, and isolate the cytotrophoblast. And we can keep them alive in petri dishes for about 2 weeks and do transport experiments with them. So there's still some of them left. The syncytium continues to form all the way around.
What did you call it?
Syncytium, the syncytial trophoblast, all the way around our embedding structure here. The trophoblast now will send cores of cytotrophoblast into each of these finger-like branches. These are going to become the villi. And some of these branches are now actually finding the maternal blood vessels. They will form smaller branches. And this fetal connective tissue is going to form blood vessels and fibroblasts and so on. So remember I said that the maternal tissue was called decidua? So that decidua that is on the deep side of the, that decidua that is on the deep side, away from the lumen, so on this side is known as decidua basalis. Again, it's the maternal tissue, the stratum functionalis, that is below the embedding embryonic tissue and will be the maternal part of the placenta. It will separate with the placenta when we talk about that on Tuesday. So this is the decidua basalis, where we have the maternal blood vessels. There is a little bit of decidua of maternal tissue between the top here and the uterine lumen. That little bit of decidua is identified as the decidua capsularis. Okay, remember, decidua is maternal tissue. The third type of decidua, remember that uterus that I drew earlier? And then we had that growing fetal tissue coming out here. So here is that, what I've drawn on the board. This would be the decidua basalis. Up here, just under the lumen, would be the decidua capsularis. Everybody kind of with me? This is growing out. All the rest of this stratum functionalis, not directly associated with our developing fetal tissue, is known as the decidua parietalis. So we have three maternal tissues known as components of the stratum functionalis. Becoming an intimate part of the placenta, inferior to this tissue is decidua basalis. That's where our multiple branches of fetal contact with the blood are formed. A thin layer above this, the decidua capsularis, and then everything else of the uterine wall is decidua parietalis. So decidua is maternal. Okay, it's that connective tissue of the stratum functionalis. The fetal tissue that develops that is not part of the inner cell mass is known as the chorion. So this component of the syncytial trophoblast and the branching cytotrophoblast is known as chorion. Where it forms the long finger-like structures, it is known as the chorion villi. Some of you may have heard of chorionic villi sampling. So I had an amniocentesis, actually I had two because I had twins. So when I got pregnant, I was 38. If you're over 35, they want to check the genetic health. And so they'll take a sampling of fetal cells that are in the amniotic fluid. If they're concerned, all right, about some of the genetic developments, and they want to do that, it's too early, there's not enough fluid to go in here and take a sampling of that before 12 to 14 weeks. But they can go in and take a snippet of this tissue outside the amniotic sac, and that's called chorionic villi sampling. So that's what they'll do early. So that's the chorion villi, but notice up here, next to the decidua capsularis, there's not much room, there's no blood vessels there, and so the chorion did not develop into a villi structure. Here's the chorionic villi. Here, there's very little organized development, so it's called the smooth chorion. So chorion is fetal tissue, decidua is maternal tissue. So chorion villi here, up here, it's known as chorion levi, and it can be spelled like that, or like that. And it means smooth. So two fetal tissues, chorion levi and chorion villi, three areas of maternal tissue. Decidua basalis, decidua capsularis, and decidua varietalis. The actual placenta is formed of both fetal and maternal tissue, chorion villi and decidua basalis. So we'll repeat this on Tuesday, kind of bring everybody back together again, and then we'll follow that through and see how the chorion villi and decidua basalis complete the formation of the placenta. And then